Namaskar. Welcome back to Introduction to Microorganisms. Let us start with our first module, that is Bacteriology. In this module, we are going to study these 7 major points, that is introduction, size, shape, bacterial appendages, bacterial growth, bacterial reproduction, their different classes, domain bacteria, and finally archaea. We are starting with the first section of this module, that is introduction. We have discussed this in the last introductory part, that is what is an essential cell, which should have a membrane which should separate the internal part of the cell with the external environment. Mainly here this is with respect to plasma or cell membrane. In eukaryotes it is mainly having a membranous organelles. It is having genetic material in the form of DNA as a genetic blueprint within a membranous nucleus, and protein synthesis is carried out by ribosomes. This is a basic or as an essential cell. When the same thing is studied with respect to a prokaryotic cell, in this image you can see that the TEM, transmission electron microscopic image or transmission electron macrograph, can be seen, which actually shows the actual bacterial cell structure. This is having inclusions within the cell, also a flagellum, a cell wall, glycocalyx, a nucleoid, cytoplasm, ribosomes, and cytoplasmic membrane. This is the basic structure of a prokaryotic cell. How do bacteria differentiate from eukaryotes? They are usually haploid for a gene, that is, they have a single set of gene structures. Loss of function is not masked by a second allele. Allele is a secondary unit of gene which is usually present in eukaryotic cells. Here it is all in a single haploid structure, hence the secondary allele loss is completely not seen here. Genetic experiments involve transferring genetic material, either not setting up crosses, although they can be mated. This is specific for prokaryotic nature. There are 3 major mechanisms of genetic transfer which we will study in our further modules. When we consider prokaryotic versus eukaryotic cell, prokaryotic cell is not compartmentalized, or there are no membranous organelles, and these membranes or cell membranes lack sterols, example cholesterol. These usually have single circular chromosomes. These are having 70S ribosomes, having subunits 30S and 50S, while in eukaryotes it is usually 80S ribosome. As you can see in these 2 images, prokaryotic cell and eukaryotic cell have different structures altogether. Although size-wise prokaryotes are too small with respect to eukaryotes, you can see there are major differences between the 2 types of cells. Prokaryotic cell have majority of organelles absent, while eukaryotes have all the organelles which are membranous and organized within the cell. These eukaryotic cells are seen in protists, fungi, animals, plants, and all these higher organisms, while prokaryotes are usually bacteria or archaea. So when we consider prokaryotes versus eukaryotes, eukaryotes are 10 to 100 micrometers in diameter. They are bigger. This is their usual size, or this is their general size. while prokaryotes are 0.2 to 2 micrometers in diameter, and they are relatively smaller than the eukaryotic cells. Membrane bound nucleus and nucleolus is present, while unbound nucleoid is present in prokaryotes. Complex appendages are present in eukaryotes, while simple appendages can be seen in prokaryotes. Linear DNA with histones in eukaryotes, while circular DNA in prokaryotes. Membrane bound organelles, whether it is Golgi complex or simple lysosome, everything is membrane bound with membrane receptors, while in prokaryotes no membrane bound organelles are seen. Organelles like ribosomes, which are non-membrane bound, can be seen in prokaryotes. In eukaryotes, mitosis can be seen. In prokaryotes, it's binary fission. No membrane receptors are present in prokaryotes within the cell. Cell wall simple when present in eukaryotes, while cell wall is a complex thing in prokaryotes. Cytoskeletons are present in eukaryotes to keep that big cell shape intact, while in prokaryotes usually cytoskeletons are absent. Big ribosomes, ATS ribosomes, small ribosomes in prokaryotes, that is 70S ribosomes. When we see this Venn diagram, we can see that there are many things which are common to both the cells, like flagella, plasma membrane, cytoplasm, ribosomes, and DNA in nucleoid or chromosomal apparatus. Also cell division, whether it is binary fission or mitosis, there is some kind of cell division usually seen in prokaryotes and eukaryotes. These are some of the basic similarities between the two types of cells. So in prokaryotic, that is primitive nucleated cells, the two kingdoms are eubacteria or true bacteria, Archaebacteria or archaic, that is oldest organisms on earth, live in these, as we discussed last time, these live in extreme conditions. When we consider bacteria versus Archaebacteria, bacteria have peptidoglycan or murine. Archaebacteria, which were discovered later in the evolutionary cellular studies, they are named as pseudomurine. They are not exactly like murines present in bacteria. In bacteria, muramic acid is present in its cell wall. In Archaebacteria, no muramic acid is present. With respect to bacteria and archaea, the 16S rRNA sequence is extremely different. So this is one of the differences between the two. In this particular structure, in this particular image, you can see among eukaryotes and eubacteria and archaebacteria, eukaryotes are further away from the two types of domains, and those two together will form the eukaryotes. This is what the scientists think is the present scenario of cells, that is their evolutionary significance. Archaebacteria, in their cell membrane and everything, they are similar to eukaryotes, while eubacteria are different. While eubacteria are present either in the form of mitochondria or in the form of primitive nucleated cells like chloroplasts within the eukaryotes. Hence, there is a lot of intermingling among the cells. Hence, this whole image is a complex study of cells. Hence, it is important for us to study eubacteria and archaebacteria to understand the evolution of eukaryotes, which culminated in evolution of human beings, that is us. We have to see how these specific cells have evolved. We have to study them more because we have studied them very less. Around 0.5% of them have been named till now by scientists. So all of these still needs to be studied. Hence, Our further modules will help you in understanding the bacteria and other microorganisms in more detail. In this section, termed bacteriology, for bacteriology is the branch and specialty of biology that studies with morphology, ecology, genetics and biochemistry of bacteria, as well as many other aspects related to them. In coming sections, we will understand the bacteria in more details. Thank you.